There are similarities and differences between DNA and RNA. So when we look at the monomers or the nucleotides, there are similarities. They both have a pentose sugar. There are 5 carbons in that sugar, and we need to focus on the 3' sugar and the 5' sugar because it's the 5' end that has the phosphate attached. We also have a complex nitrogenous base, which we consider the letters of the code, because when we talk nucleic acids, we're talking about codes. So we have a 5' and a 3' end. When we start talking about similarities for the entire nucleic acids, they are covalently bonded, these sugar phosphate backbones, together. And it's the hydroxide hanging off the 3' end that the phosphate will attach to. So ultimately, the entire nucleic acid is a linear structure that has a sugar phosphate backbone, and it has directionality, which means that one end is going to be considered our 5' end and the other end is our 3' end. They are also going to have perpendicular nitrogenous bases coming off that backbone. So as we start looking at these, if we zoom into the differences, RNA has ribose sugar, DNA has deoxyribose sugar. So it's still pentose, but there is one less oxygen in DNA nucleotides. They both have cytosine nitrogenous bases. They both have guanine nitrogenous bases. They both have adenine nitrogenous bases, but DNA has thymine and RNA has uracil. So it's this area right here, if you see T or U, that you'll definitely be able to tell the difference between DNA and RNA. Again, when we look at this, we often think of RNA being a single strand and DNA being double-stranded. We can also see this is RNA because of the U, and this is DNA because of the T's. So when we start thinking about DNA and that double-stranded structure, the other thing we have to think about is that it is anti-parallel. So we have 2 strands and they run parallel to each other, but they are in opposite directions. So we can see the 5' end is on this end and the 5' end is on that end over here. Now, whenever we're talking nucleic acids, we kind of need to think about genetics, and that central dogma of genetics is that DNA goes to RNA through transcription and RNA goes to protein through translation. Now, it's some of these extra differences in their structures that cause differences in their functions as well, which is a big theme of this unit. So, RNA does translation through messenger RNA, transfer RNA, and ribosomal RNA. And there's even small RNA that actually shut down this process. So again, we have DNA hanging out up in the nucleus of a eukaryote that's storing the genes. From this, we can make messenger RNA. That messenger RNA can have a cap and tail put on it. It can get spliced into mature messenger RNA, which then would leave the nucleus, find a ribosome, and at that ribosome, we have ribosomal RNA, and the transfer RNA is connected to amino acids, which bring them to the ribosome. At the ribosome, the ribosomal RNA, which is sometimes called ribozymes, as in enzymes, will attach the amino acids to one another. And again, this isn't our whole process on genetics, but we have different types of RNA. It's not just one type of RNA, and ultimately they work together to ultimately make the proteins. So there are similarities and differences that you definitely need to know about DNA and RNA.